Hello everyone, welcome to 2-minute lessons, and I will continue to talk about meiosis part 2 in this lesson. Before we proceed, this is what you need to know. Chromosomes are tightly coiled segments of DNA that contain genetic information. Most people on Earth have 46 chromosomes, which are divided into 23 pairs. These 46 chromosomes contain all the instructions to make you, or anyone for that matter. It's why you have a certain hair color, eye color, height, weight, skin, tone, or a different voice. In sex chromosomes, if individuals are having two X chromosomes, they are female. Individuals having one X chromosome and one Y chromosome are male. In lesson 6, I have explained about meiosis 1 and all the substages in prophase 1, namely leptotene, zygotene, pachytene, diplotin, and dikinesis. You can go back to that and refresh. Let's move on to other stages in meiosis 1. With that information, let's see what happens in meiosis 1, that is metaphase 1. This is the second stage of meiosis 1. Homologous chromosomes, 2 pairs or 4 chromosomes gather along the equator of the cell and prepare to separate. Next, the spindle fibers attach to the centromeres of each chromosome. Take a look at that image. Both sister chromatid pair are turned towards the same pole. Chromosomes form two parallel plates. This is the major difference between meiosis and mitosis. It causes the two members of each chromosome pair to be separated from each other during the next stage of meiosis, that is anaphase 1. During this stage of meiosis, the cell starts to lengthen. The two homologs of each chromosome pair separate and move towards opposite poles drawn by spindle fibers. In meiosis at this stage, the chromatids remain together as one complete replicated chromosome. In meiosis 1, the next stage is telophase 1. At each pole during this stage, there is a complete haploid set of chromosomes. But each chromosome still has two sister chromatids. A cleavage or furrow appears, and by the end of the stage, the parent cell has divided into two daughter cells, and the nuclear membrane begins to form. Cytokinesis, it is the division of the cytoplasm as opposed to karyokinesis, which is the division of the nucleus. It begins as the cell starts to lengthen during anaphase and ends when the separation of the two daughter cells is complete at the end of telophase. Division of the cytoplasm occurs in both mitosis and meiosis. Daughter cells begin immediately to prepare for the second meiotic division. Prophase 2, the first step of meiosis 2, begins with the two daughter cells produced by the first meiotic division. Take a good look at the image. As in prophase 1, the chromosomes are condensed. The spindle fibers move the chromosomes to the middle of the cell. This stage is similar to mitosis, but remember, here there are no homologous chromosomes. Metaphase 2, chromosomes line up in single file along the equator of the cell. This is in contrast to metaphase 1, where chromosomes line up in homologous pairs. Anaphase 2, the chromosomes are pulled by the spindle fibers and the chromatids move towards the opposite ends. Important to note that the chromatids are not identical due to crossing over and exchange of genetic material in prophase 1 stage. Telophase 2, the chromatids are at the opposite ends of the cell. Slowly, the nuclear membrane forms, and you can see two distinct nuclei inside each cell. Now the cells is ready for the final stage, that is cytokinesis. Cytokinesis of each cell occurs and form 4 genetically different cells, each with half the number of original chromosomes. The genetic recombination and segregation is said to provide genetic diversity to the offsprings. Here is the entire sequence of events in meiosis 2, where sperm and egg cells form zygote, undergo reduction division, recombination, and finally segregate to form 4 daughter cells. Hope this lesson was interesting and easy to understand. Try learning the changes in each stage by observing the relevant images. Thank you for watching. See you in my next.